Laughing now.
Okay. Well, welcome back, friends.
Hey, thanks.
Okay, so if we can just kind of recap where we left off to get our mind back into thinking about human brain and senses. So what did we decide our anchor phenomena for the unit was going to be?
We had said that we would use what causes color blindness, your eyes, or how your brain interprets what is being seen. Because we knew that we needed to connect to more than just the brain and sensory details, but we needed to connect to the cells, so we thought that was a way to do it, and that ties into our power standard, which is MSLS1 8-8, which is gather and synthesize information that sensory receptors respond to stimuli by sending messages to the brain for immediate behavior or storage as memories.
And then we put in here a couple pictures that we were going to show the students. Were we going to show both of them, or we did talk about that, right? We're going to show one and do I notice, I wonder, and then show the second one of the different colors of what one is what people without color blindness see, and the other is what color blind people see, and see what they come up with without telling them. Okay, good. So I think we left off on lesson 4.5, and we had that 0.5 because this was a us created lesson, right? Not in the FOSS kit. Okay, so Jenny, I think you had a good idea for phenomena. What was that?
For the lesson level phenomena we're going to do for this one, because this is where we change into talking about the cellular level. The students haven't had a lot of experience with cells at this part in their education, so we decided we're going to do a vinegar egg, which is where you put the egg in a regular egg into vinegar. And let the shell kind of soak off over a couple of days, and then when they look at it, they're going to see the inside of the egg intact with the nucleus on the inside, which is the yolk, and actually see what a single cell looks like at a modified level, magnified level, so that it'll just draw in their interest to figure out, like, why does it look the way it is? How is it that, the parts and pieces of it to lead into that. So
were we thinking too, kind of like those pictures, that we would start with just the vinegar egg and have them do I notice, I wonder, and then pose the question?
Yeah, so we'll just show them the egg, walk around with it, maybe even let them touch it gently, to see what it looks like and feels like, but not tell them anything about it other than, what do you think this is? Kind of lead them into the ideas and have them do the I notice, I wonder process with that, so that they can really get their, curiosities out. And then we'll lead them into telling them that this is a single cell, and then start to break into the lesson.
So kind of our phenomena question is, did you know this is a single cell after they look at it? And then probably in the end they'd have to provide evidence to prove why that was a cell.
Yeah.
That they could see. Okay, and then you can bounce it on the table. They do bounce like a bouncy ball. Yeah.
And somebody will break one.
Right, that's the whole point. Do it at the end, Mr. Easton. Okay, and so what does this lesson, what are the content focus, what do we want the students to take away from learning about a single cell?
We want them to know we attached this to MSLS1-2, which is a supporting standard, and it talks about the different parts of this, like the structures of the cell, not necessarily the functions of those, but we will address some of those within the lesson. But so we want them to know the parts of the cell, some of the functions, but we will not assess that specifically, but we do want to give them a basis for that. We want them to be able to create a model of animal or plant cells, to be able to show sort of the way that looks and how it works, a cell. And then we're also going to do like a comparison between a 2D and a 3D model in some fashion with the students as well.
Okay, well, great. Then we'll carry on to our next investigation.